Hello and welcome to this webinar, How to Escape from a Cytotoxicity Failure. My name is Lise Vanderkelen and today I will present this webinar together with my colleague Audrey Turley. Hi Audrey.
Hi Lisa.
How excited are you?
I love talking about cytotoxicity, so I am very excited today.
Is this something that happens a lot, cytotoxicity failures, or?
It's very common.
Yeah. From my experience, it's when this happens in the lab, our sponsors are most of the time in a panic. They don't know what to do. It's a burden. You need to. They ask all these questions. They think that their device is not good. So it's, for me, I feel it's a painful process. I'm not sure what your thoughts are in general.
I feel like it can make our sponsors feel like they stop before they even get started with their biocompatibility. So I'm excited to talk about how they don't have to have that happen today.
Yeah. So what we want to do is we want to make this burden, we want to make this painful event something more fun, something that they can escape from. And when I think escape, I also think about something fun, which is an escape room. So in an escape room, you're locked with your friends, your family, your colleagues. You're all together in this room. It's a great team building. And you need to find clues in the right order, try to make some puzzles, and then you get out on time because you have solved the mystery. So I thought that we could make it more fun together with you to make the cytotoxicity failure into an escape room adventure.
Very exciting.
First of all, before we start playing the game, we have to understand why do we perform the test for cytotoxicity? Why do we test medical devices? So it's part of the biological safety evaluation. It's described in part ISO 10993-5, but also in the general safety evaluation, ISO standard part 1. It is an endpoint which you need to evaluate. So I think, I might be mistaken, but you can correct me, that 99% of all devices on the market have been tested for cytotoxicity.
Yes, or they should have been. Every medical device has to have a cytotoxicity evaluation.
One point you need to evaluate through testing, or it's mostly done through testing. It's a quick test, it's cheap, it's easy to do, it's in the lab, so there is no reason why not to do it. And some of our customers also use it as a kind of QC control testing of their manufacturing process to make sure that everything is still under control. Whether the cytotox test is the best test to select for this, we can discuss about that, but that's out of the scope of today. And of course, it's also a test which is used as an incoming control to check new materials, to check components that you always use. So it's often used, and because it's so widely used, you also have some failures. Now, in general, the ideal scenario, what everybody has envisioned when they start a test, you take your device, you extract your device with the minimal essential media, expose the extract to the cells, the mammalian cells, and then you look for cell viability. And what you want to see is happy cells, alive and kicking, and it's a pulse. You can see this brownish color. This is actually a picture under the microscopes of the cells that are happy because there is no toxic component present in the extracts that kills them. Now, we're not going to talk about this scenario because this is what we all want. It's a clear path. You go through, you can go to the next phase. But sometimes, or more often than we would like, we do the device extraction, expose the extract to the cells, but all of a sudden we see a failure. That means that cells are dying, are not happy with the content, are actually, that is a mystery, or that's the burden, and we want to create this into a mystery to solve today. And think about these black dots. That's your escape room for today. So we want to find some clues in this escape room. Now, of course, I think you as an audience are thinking, okay, this is not how I envisioned an escape room. So if we all close our eyes just for a second, open them now. Ta-da! Yes, this is the escape room that we now are going to play with. This is actually your cytotoxicity failure, but in a more nicer way. And this is where you enter in the room with your colleagues, with your team members, to try and solve the mystery. So now today, Audrey and I will be locked up together in this room. I'm cheating a bit because I'm using Audrey's expertise over 20 years, I think, if I'm not mistaken, in dealing with these things. So I hope I can get out. And in this room, we have hidden 6 clues. We have to find together the clues in the right order. That's essential in every escape room to get out of the escape room. And that will help you as well. So if you follow these steps, you can also escape at work when you're dealing with this failure. So let's look for the first clue. Just looking. Yes, the easiest one, the first clue is right on the floor. It are the footprints. And every time you're locked up in this room because of a cytotoxicity failure, you should ask the first question, are the results that I've received, is this fail really a fail? So you have to have confirmation of the validity of the test results. So what you're going to do is you're going to look at the report you have received, and you're going to see, okay, does this data make sense? How can you check it? You check, okay, what lab facility did it? Is it accredited? Is the test accredited according to ISO 17025 or under GLP? So was the staff trained? Was the equipment qualified? You look at the test controls. Were they okay? What were the conditions? And so on. Were there expired product use? These are all very important questions. And I think you should doubt at the beginning, am I in the right room? Should I be entering here? I'm not sure, Audrey, what are your thoughts on it? Should you question the results?
I think as scientists, we have to always say yes to that. Not that as a lab, we think it's often that this comes up, but it's the first starting point. Why go down the road of investigating a failure if you're not confident in the data? So always start, verify the data, make sure it's correct.
Yeah, indeed. And that's very important. Verify the data, ask the lab to look in some things. It's not the nicest thing. I've experienced this as a lab manager often. And it can happen. What can go wrong during testing is that you have an infections of your cells, which gives odd results. Your negative control cannot respond as you want. You want to see 100% viability and you see some dead cells, or your positive control is not acting the way it should be because it should be a very cytotoxic product. Normally, if you have received a report from a lab, this should have been observed by the lab before. Another thing, what a lot of our sponsors do not know is that a cytotox test is performed in batch. So we do a lot of cytotox testing simultaneously. Therefore, if something goes wrong with the complete setup of the testing, all of our sponsors would be affected. And that is something that I've not experienced, that all of a sudden all the sponsors would respond like say, hey, there's something wrong. And that also gives you some guarantee that the testing is under control. And that can also be added in your root cause investigation. When you're in this room checking the validity of the results, you're saying, okay, 100 devices were tested in the same run and only mine is not doing what I want. I am seeing a fail. Can also mean that the testing is under control. What I also finally want to add is take time to evaluate this, but not too long. Don't stop here. It's easy to say, okay, the results are wrong, but you have to continue and move forward at a certain speed. Don't stay here too long, but take the time to make sure that you're locked up in this room, that you're here for a great team building, yes, but you want to make sure it's the right time for it. So the first step in how to escape is very simple. If you have confirmed that there was a laboratory error, ideally the lab also confirms that it was a lab error. That's the only way how to validate an error. I don't think as a detective you can say, oh, it's based on my gut feeling that it's not okay. That will not work. You can retest, and if that passes, you are out of the room and you have to find another way to do team building. You have escaped. Your assessment is complete. Again, you have to have scientific proof, I think, Audrey, to confirm that it was a lab error.
Absolutely.
Yeah. So let's go next. The first clue, very simple, very easy. I think we can all do this at home or at work. So if there is no laboratory error, not at all, you have confirmed it, what do you do next? You have to look for the second clue. I'm not sure, Audrey, can you see it?
Yes, I think we want to keep looking, and the next clue I can see is on the couch. So let's walk through it, and I want to add to what we found in the first scenario where Lisa said, don't stay there too long with the lab failure. And you may say, oh, but I know that this should not fail. We're going to give you lots of examples of that exact thing happening and ways it can cause failure. So hopefully stay with us. We're going to get through this. So in the second clue, we're going to talk about a few more things in the testing we want to look at. So this is not necessarily a lab error, but maybe something we should have done differently when making decisions about how to run the test. So this is talking about test parameters. So we're going to first talk about extraction time. So this is the time that the device is extracting in the minimal essential media that Lisa showed earlier, so MEM. So we can do typically 24 hours or 72 hours. And you can see here, we make that decision based on the contact of the device with the patient. So prolonged, like a permanent implant, would of course be extracted for 72 hours. Something that's used less than 24 hours, maybe for 6 hours or so, would have a 24-hour extract. Now we have here listed a 4-hour extraction, and that's for, we want to think about, we know there are their device is only used for maybe 5 minutes, but we're going to try to extract them for 24 hours. But Lisa, can I just say, I immediately want to go to 4 hours because that's exaggerated enough? My device is only used for 1 minute.
Ideally, I would like to do that. I would say my device is only used for 1 minute, so 1 hour, 3 hours. I hear you laughing. From my point of view, I would say I could avoid a lot of cytotox failures potentially by just reducing that. But yeah. I know, and I'm sure, and that's the answer. This is not allowed. So you can do this only, and I think 4 hours is the minimum extraction time. Yeah. I don't think you can go lower according to the ISO or what regulatory expectations are. You can go lower, but only after you have a clear fail after 72 or 24 hours, and then you can justify and say, okay, 4 hours because there's only 2 minutes contact time, whatever. But that's part of a justification. So don't go it, although you would say like, I think it's okay. It sounds scientifically correct. 1 minute, 4 hours extraction, perfectly worst case. That's not the reality.
Yeah, that's absolutely true. It's like, so the regulatory agencies want to see the failure. Yeah. They want to see it at the exaggerated conditions they expect, which is at a minimum 24 hours. And if it does fail, then you can say, oh, but my device is used here. Let's try a lower time.
So to summarize, first fail, then you can go lower, but not direct.
Yes, correct. Absolutely. Okay, so let's think about another part of the test parameter we want to consider, which is an extraction ratio. So when we prepare samples, we have to determine the ratio of sample to the fluid, to the extraction fluid volume. So the two main options used here are 3 centimeters squared per mil or 6 centimeters squared per mil. And you can see by this table that we have from the ISO 10993 Part 12 standard, which is all about sample preparation, that we determined that ratio based on sample thickness. So perhaps there was instruction to test the sample at 6 centimeters squared per mil, but once we go back and verify, we realize, oh, the sample really is thicker, so we should have done it at 3 centimeters squared per mil. So that's an example of this not working out correctly in the lab.
Yeah, and but if I understand correctly, then I would, okay, I look at the thickness, and if my device has a part that has only 0.4 millimeter thickness, then I just take 6 square centimeters per mil as the extraction ratio. So the table is very clear for me. I think that's worst case.
Yes, very clear. So one other condition though, and maybe some of you guys are already asking that, is, well, what if my device has different thicknesses? Because it's a complex device, different components. So let's talk about a scenario for that. So we had an example of a device which had different thicknesses. So this is just a pretend device that we're talking about today, where the IV bag is greater than 0.5 thickness, but the rest of the components are less than 0.5 millimeters squared. So we did a worst case scenario extraction using the higher ratio, and we got a fail. So, which we were following 10993 Part 12. Here's a footnote from the standard that tells us, hey, one thing to consider is doing the worst case scenario. If you have a mixed thickness device, go from the thinnest component and use that ratio. Well, in this case, it was over exaggerated because when we look at the surface area for the specific device, the greater surface area that was thicker consumed more of the surface area of the device. So from a ratio perspective of the whole device, we were really exaggerating that extraction. So what we did is we separated out the components based on thickness, and then we received a passing result. So one of those situations where you expect it to pass, but again, just thinking about it in a little more detail. Yeah. Okay, but we have more parameters to consider. Did you have something to add on that one, Lisa?
No, I think it's very important that you just not always take the worst case, because we have seen some failures like that, and they only take the smallest thickness as the worst case, and then you have a failure. And ideally, you can already avoid that by doing the calculation. You don't have to go like with the other example, go first to the worst case overall extraction ratio. You can actually use the correct extraction ratio. I think regulatory there is fine.
Yes, you can use that justification upfront to talk about why you prepared it that way. Exactly. So another parameter we want to talk about is the extraction solvent. So far we've only talked about extracting an MEM fluid, but there is an option to use 0.9% saline. There's other alternates, but really MEM solution is used, usually supplemented with a calf serum or not, and then the saline. And so it's nice to think about if your device is only ever exposed to saline, like an IV bag, we're talking about those a lot today, and it doesn't make sense to extract it in something else, especially if we had seen a failure before, but it's only ever exposed to saline. Let's use an appropriate clinical solvent there. And another thing we want to think about in the parameters is how the device was extracted versus how it's used clinically. So again, thinking about an IV bag, this gives us all those perfect situations we want to talk about. If we immerse an IV bag, we would just cut it up and put it all in fluid, inside and outside. But that's not how it's used or how the patient will be exposed to it. So it makes sense to only extract the inside portion. And in this case, we would also use saline if this was a saline-only IV bag. So just an example of when those things could be considered, so it relates clinically to the use of the device. The last thing to think about test parameters, now you may think this doesn't relate because it's not test parameters, but it's important to understand how the sample was manufactured beforehand. So thinking about the sterilization method. So those can impact the device and the cytotoxicity test. So if the device is terminally sterilized before a patient is exposed to it, we need to capture that step in the cytotoxicity test, but that can also impact the cytotoxicity test. So I'll give you an example where we had a product that was being sterilized with hydrogen peroxide. Now, typically hydrogen peroxide leaves a water residual, but we have found out that it will leave very, very small amounts of hydrogen peroxide at a level high enough to cause cytotoxicity, but not necessarily high enough to be detected in a quantitative test. So that tells us that the level that's left on the device may kill the cells in this test, but would not necessarily harm the patient. So we have to then think about that from a patient safety perspective, but we need to understand the sterilization step used and consider that that could be impacting the results here. Hydrogen peroxide is a safe sterilization method, but we still have to think about what's left on that device and the exposure to the patients. I think,
yeah, what you also need to do, yeah, is then when you have well described all the parameters for the testing, but what you also need to do when you're in this phase in the room, you have to dig into your historical data. You have to look, okay, what were the parameters that I used before, and that can give you a clue whether or not did you change something, and that can give you an indication. And you also have to look at the type of method. So as some of you may know, but we have not discussed this in detail, and we're also not going to do that, but there are different types of cytotoxicity tests. You have qualitative, quantitative tests, and they give you a cytotoxic response, yes or no, but they are, from a scientific point or how the test is built, they're a little bit different. So in general, the dye exclusion is a qualitative test. It's done by visual inspection. It is a more subjective method, and it's less sensitive as the quantitative XTT or MTT method. So if, for instance, now you have a failure and you see, oh, and in the past it always was successful, but then I used a qualitative method and now I'm using a quantitative method, that could be indeed the reason, because the quantitative method is more sensitive as the qualitative. But you cannot, and that's very important, not say, okay, I just ignore the fact that I used another method, I ignore this failure, and I just switch to a method. Or you say, oh, I want to use another method to see whether there I have a pass. So I think the most important message here is look at the data, see what was different from your historical data, but never just, let's say, ignore a failure and say, I'm switching to another type of cytotoxicity test. I think, Audrey, I'm not sure how much we can emphasize this, but I think you can add this as well.
Right, we don't want to just test it to compliance.
Yeah.
If we see a failure, let's understand it and move on from there. It's not that a fail here means, you're permanently punished forever. We can find other options.
Yeah, but the option is not just ignore it and switch to a test that gives you.
No.
That would be considered as testing into compliance. You're getting out of the room, but it's not the right door. You have to leave out front. So it's not okay. So really important message, take home message. Don't do this. So to capture our all the summary of the first two clues. So if it's not a laboratory error, look at the parameters. If you have not selected the suitable extraction parameters up front, you can retest with the updated extraction conditions sometimes, you need with the 4 hours extraction and justification, and then you're almost out, right, Audrey? You cannot just say
almost.
Almost. So it's like, no. So you see, there is an error here. It says, okay, you have to pass or go by a biological risk assessment.
Yes, so this lets you, so if you found your root cause in one of those first two steps, you automatically get a jump to step 6, which is great. That's like your shortcut to escape the room. But before you can escape, you have to have this passport. So this is written documentation, and we're going to talk about this at every step, and not in detail, but to highlight that you need to write down what you're finding out. So this is a great tool to use, not just for documentation purposes for your submissions, but also to track what's going on historically, so you can keep track of that as a company. So first thing we're always going to talk about is device intended use. So we talked about that at the beginning. How long is it contacting the patient? Is it used with only saline? Does it have only contact in the fluid paths? You can hear us getting those details ironed out. You would do that right up front in your risk assessment. And not from the perspective of how the device functions as far as what it does for the patient, which is important, but in this case, that's a side note to how it contacts the patient. So that detail is really critical. And then, of course, we're going to talk about any testing results, all the failures too, because those are teaching us, plus what we've discovered through additional testing. Then we're going to talk about the historical results as well. Lisa, will you forward the slides for me? I seem to have lost control. There we go. It's coming. Thank you. So historical data, like Lisa talked about, and that's not lab historical data, right? Again, that's the customer, that's your sponsor, that's you guys, what you've done as a company in the past, keeping track of all of that data. And then, of course, we'll talk about this later when this becomes important, but material information or chemistry, this is something like the hydrogen peroxide residuals, that would be discussed here. And then our last two components are, what did we find in that investigation? And then what is the risk to the patient from those results? So I just want to give an example with the hydrogen peroxide, if I could quickly walk through that so you could see. We would talk about the device use. We showed an example of an implant here. And so you would talk about the contact. That's a pretty high-risk device, so we want to make sure the exposure is low enough to ensure patient safety. We would talk about our results, our history. We've always done hydrogen peroxide sterilization. This is not new. And the investigation we did with hydrogen peroxide was it was low enough concentrations to cause cytotoxicity failure, very low, so low that it almost didn't show in our chemistry testing, and the amount that we're getting off won't cause a risk to the patient because we're going to provide a lot of historical information on that as well. So that's our investigation, our justification, really tying everything together in a nice written document that fully supports patient safety based on these results.
Yeah.
And with that,
with that,
we
you
escape. Yay, we got out.
I think it's very important to say you need a passport to get out. That's what you need to get out of the room. Only after laboratory error, that's the way out. Without a passport, otherwise you need a risk assessment. And I'm lucky today in this escape room because I locked myself up with an expert, so we can easily, right?
We will get out together.
That's for sure. Now, of course, this doesn't end here. Right? You have, of course, it's possible that even a retest with the updated extraction condition, you still have a fail, or from the beginning with your original fail, all the extraction conditions, the parameters were fine, were okay for your device. There was no justification or no reason to actually make them less stringent, less or less worst case. So that's also an option. And then, of course, I think we all know the game and try to see it. We have to find for the third clue. So we have to ignore now the footprints. And there it is. Some blood on a...
Hidden. That one's hard to see.
Yeah, that's one. They will become harder and harder. So the third clue, what are we looking for? Now, we have more looked at the test parameters. Now we really have to look into the device's materials, its components, because we know some materials, they cause a cytotoxic response, so as natural rubber, the organotin stabilized PVC, and so on. These are also our positive controls in the test. There are also metals like silver, zinc, and so on that causes a cytotoxic failure or are very likely to. Plasticizers used like DHP, and some drugs are coated with some devices are coated with drugs, and these drugs, of course, when they leach into the extract, they can also have a cytotoxic response. So at this moment, you can really be like Sherlock Holmes and dig into the supplier information, get to know your device really well. Sometimes it can be very tricky to get all the information from all your suppliers available. Sometimes it's really a deep investigation that you need to do, and we would advise you to do so. Perhaps even before you start with the cytotoxicity failure, before you enter the room, you should know this actually, but dig into all the documentation and look which of the components might cause a cytotoxic failure, which is already known. Not everybody knows it. I don't know it by heart like Audrey does, but it can be helpful then to ask with expert or material expert to guide you in this. Now, if you have found a potential component of your device that has a potential cytotoxic effect, then what you can do is you have two options. First option is, okay, you take your device and you remove the cytotoxic component out of the device, and you perform your cytotox test again under the same conditions. So you do the extraction, you expose the extracts to the cells, and if you then see a pass, you know, and you can actually confirm that that component indeed was causing the cytotoxic effect. It says also that you need a biological risk assessment to get out, but that will come later, but you have already that option. I think, have you ever done that or?
Yes, we have a case study, perfect one to talk about with this. We had a hydrogel that needed to conduct an electric current to a patient. So we were testing the hydrogel and it continued to fail. And that was a surprise to our customer, although we had seen this in the past with other hydrogels. But there seems to always be something a little bit different of what will cause the failure. We can't always say it's the same for every type of product because they're all made a little bit differently. So in this case, what we discovered is what carried that electric current to the hydrogel was a thin sheet of tin backing. Now that ended up being in the exposure, but we didn't know at the time what it was. Typically, we're testing these things blind without material information. So what we did is we were able to peel off that tin layer on the back of the hydrogel. So we were able to remove the cytotoxic component, and that tin layer doesn't have patient contact because it sits above where the patient's only being contacted on one side with the hydrogel. So that was able to help us get those passing results. So ta-da, we were able, this would help us escape the room with a risk assessment, like Lisa said, but it would help us get to that passport point. Yeah.
And then, of course, I said there is one option. There's, of course, also a second option. It is not always possible to remove the material or the component that is potentially causing the cytotox failure from the device. Sometimes it's just technically impossible. What you can do then is you can test the medical device again, but you're going to test or expose different dilutions of the extract to the cells, and you're going to evaluate at which dilution you see only again the cell viability, which is okay, and a non-cytotoxic response. And in parallel, you test a predicate device, so the device that is made of the same material or is already on the market, and you see whether you observe the same failure and pass at different or at the dilutions. And if that is the case, then of course that gives you a good justification to say, hey, my device has a potential cytotoxic component. We are aware of it. We have a predicate device that has the same components and also shows the same cytotoxic response. And if we dilute the extract, then we also see a similar pattern. I think also that I think Audrey can confirm with a very nice case study.
Yes. So this is an interesting case study because it's about a product line, actually. So different components within this product line. So we'll go back. There we go. There are some toothpaste that are being designed as medical devices for the way that they're treating, gum and oral issues. So that makes sense. But when you try to put it in a cytotoxicity test, it's going to fail for an abundance of reasons, not just one component, but just the very nature of toothpaste. This very sensitive cell culture system is not going to love that very much. So what we did is we said, okay, take your product and compare it to several other toothpastes on the market. They will have different components. Their product was more prescriptive with certain ingredients. However, just in general, to show the regulatory agencies, like, yes, toothpaste is not going to pass cytotoxicity without some dilution. We expect it to show cytotoxicity, especially because we want toothpaste to kill germs, right? So it's cytotoxic to some extent. So we ran several dilutions compared to several different types of toothpaste, popular ones on the market used by a variety of patient populations. And we were able to show similar levels of cytotoxicity out to a dilution level of like 1 to 1/16 or something like that. So this helped us to not weigh so much of the evaluation of the safety of the toothpaste on cytotoxicity. It's a nice test, but it's not the end-all be-all of biocompatibility. So we kind of wanted to, like Lisa said, find a way to not stay in this room for a very long time, but have a way to get out and move on to the rest of the assessment.
Indeed. Now I just have let's say the intent to take my toothpaste tomorrow to the lab and see what it does, just for fun.
Yes.
But yes. Still, okay. We're back. Step 1 is clear, laboratory error. If you have not selected the right parameters, it might be a way out. Then if that's not the case, we can look into the materials and know whether or not they are potentially cytotoxic, because we have literature data, we have already testing data available, and we can retest without the cytotoxic component as we discussed, or test with dilution. If we have a pass at this case, and I already talked to you about it together with Audrey, you can get out of the room, but you need the risk assessment. You need the justification, as it was well explained in the case studies. You cannot escape without this risk assessment. You take all the results together, you justify it, look at the similar devices, the predicate device, the dilutions, whatever, and use that. It is not, you don't have to write a book. You just need to have the justification. in a right wording, in a nice story. It's a storytelling that you're doing in this assessment so that everybody understands what you have done and why it is still good for you to move on with this medical device or this material. So, yes, third time, right? We're out.
Yes.
6 clues, still 3 to go. So we'll go into the room. After, of course, it's normal that we stay in the room if we haven't found a cytotoxic component, or if, and that happens also, if the retest or the dilution testing, it was not successful. If it all showed failures, then we need to look for the 4th clue. So let's see.
Yeah, there we go. There's the 4th clue. That one was sneaky too. So now this is a lot, again, of work done in the manufacturing facility by the customer. So really thinking about what is my process. And I would say, if you're automatically saying, my process hasn't changed in 10 years, I would say you probably need to go and ask questions about what has actually changed. It may not have made it on the level of somebody saying, oh, this is an actual change. So we're going to talk about some of those today. To help you ask really good questions, Lisa and I are going to make you guys really good investigators after this today, not just for cytotoxicity, but for everything. Good question.
It's a great team building. I told you it's a great team building. How to do all the little stuff.
It is for many reasons. So the first thing we want to understand is if there's any supplier change. One thing to note is, especially for things like polymers, one polymer, the same polymer from 2 different suppliers does not make the same polymer. We know that there are proprietary formulas, other additives put in there that they are not releasing. So even if they start with the same name of the raw material, if it's coming from a different source, they're doing different things, that makes that supplier material different. So, looking at that very closely to understand what in that process has changed. And again, this one is tricky because I think there are changes that happen that don't get documented as changes. So this is a lot about change control in your own company and what you guys are monitoring as changes. That's hard with suppliers because you don't have control over that always, but even it can be hard within our own walls of our company.
Yeah.
And so something to really think about, ask good questions about that too. I have a good example of this about process change. So we had a company that was moving all of their equipment from one site to another site. They were expanding, they needed a bigger facility. Everything was staying the same. And we said, are you sure? Even your suppliers? Yes, everything was staying the same. Even the pieces of equipment were the same. They moved to the new facility and their devices failed cytotoxicity. We said, just run cytotoxicity as a quick check. They failed cytotoxicity. So we said, okay, let's look at this new location. And we said, maybe test the same device, test the device from the old location, new location. Let's compare them at the same day in testing with the same cells. Let's see if it's, something weird with the lab or whatever. No, they still failed from the new facility. So we did some investigation and we found that there was a residual that came off from the new device. We ran some chemistry because we really couldn't figure out where this was coming from. And it turns out that the residual that we found is a chemical that's used in surfboard manufacturing. Guess who their new neighbor is at their facility? They share a building, just different, separated locations within that building. So there was a problem with the air ventilation system, actually. So the solution there was to correct the air ventilation so they would have separate systems. So just, again, this manufacturer would say, there's no reason for this failure. Maybe they would say the lab is doing it wrong, right? So this is why it's good. Ask lots of questions, be very curious, because we're always surprised, I think.
I think it's a very good example to see if you stay too long in the lab phase, and focus on it's a lab error, which can always be the case, by the way. But move on, and then it is a small thing, this is a very great example, a funny one, perhaps not for the original.
Yeah, Maybe not so much for the sponsor, but it was we all learned a lot. Our eyes were open with that. So, okay, we also want to think about products used within the process. And maybe there was a product change, which wouldn't maybe mark a change because you're still doing the same step. And somebody's saying, well, it doesn't matter. Maybe we're making that better. We have an example where somebody said, oh, we need to clean these up a little bit because we need to address some bioburden issues. So we're going to add an IPA rinse, so isopropyl alcohol rinse to the product. And we said, okay, run a cytotoxicity test. Well, they started to fail just from that just from the IPA. So this was IPA sprayed onto a plastic. So again, just a reminder, not all the IPA will evaporate. It will stay as a residue on the product. So it was actually an easy solution that we said, why don't you just add a rinse of water after the IPA, make sure, of course, the water is clean, not contributing anything further, and that helped them pass the cytotoxicity. So thinking like, oh, we're solving one problem, you might be creating another. So again, being mindful of actual change, where it might be, even if you're saying we're just improving the process, there still needs to be those checks in the process as well. Okay, what other parts in our process? Packaging, of course. So questions again, are we changing anything in our supplies? I don't have an example of packaging affecting cytotoxicity. We've seen that impact biocompatibility in other ways, but again, it should be asked. Just because we've not seen it before doesn't mean it can't happen, just like the surfboard, right? It's possible. It's possible. Okay, this last one, thinking about a change in handling. So I have an example of this where we had a client that would use glass beads. It was part of a raw material that they would get for their device. Well, these glass beads had a lot of hand manipulation through the process. And somebody on the line said, I want to make this cleaner, so I want everyone to wear gloves as they handle the glass beads. Well, the gloves that they chose, of course, were latex gloves, which led to failures because latex is cytotoxic. So again, it was one of those things that was a change to improve the line, but it actually caused issues with the cytotoxicity. So, and maybe not even documented as a change because somebody thought, oh, I'm improving. I don't need to make this as a change, but it's a change. So again, back to all these, analyses, what could possibly be the case, asking a lot of questions. We're at step 4, asking what changes are happening in the process. So if you've identified a change, could find a solution to address that and get back to those passing results for cytotoxicity. Again, documenting that in a risk assessment, saying all the things we know about the device, all the historical stuff, and maybe improving change control in this case particularly, I think. So with a risk assessment here, documenting all of these different phases, we can escape. Yay! Again, man, we are a really good team, Lisa.
That's great. And I think here, you also, if you think that the change is causing it, you have to have proof again. In your risk assessment, you have to have data to support it. It's not just, I think it might be the water, it's just saying something random now, but you have to have the proof and demonstrate with some causing cytotoxin failure that that's actually the root cause.
Yes, and I would add to that, that's a really good point, Lisa. Don't be afraid to go get more data to show that. There's only so much, a lot of people will say, well, can't you just look something up and write about it? I need data. Data is what I write about. So we have to gather that data somewhere. So yeah.
You have to tell your story, but you have to have some input from the outside to make that story maybe good and easy to read, but also, reliable.
Yes.
So we're almost to the end. We're at the step 4. The change in your process. It can be that you haven't found any change. That is still the case. You can have documented everything. You have digged in deep. Your colleagues may think, oh my God, there she is again, or he. You have found nothing. There is still a way out. So we need to find for a 5th clue. So just go and see where it is. Okay, on the chair, we can find the hair. It's very small detail. Look closely. What's the fifth clue? The fifth clue is a vague clue. It's more something you can do. So you have done a lot of investigating, a lot of data that you have looked into. You have done some testing. Here, we're starting more in the phase of investigational testing. So you have your medical device. You have no idea whether the components are cytotoxic or not. There is no information. You have no change in your process. So what you're going to do is you're going to test different lots of your product to see, was it just an outlier or not, or what's happening? Is it to all my devices? You're going to test, if possible, of course, at different phases of your production process, your device. That can also be the case, or you're going to test the different components of your device. So you're going to do that through testing. So literature is not helping anymore. Change control checking is not helping anymore. The ISO standards with extraction conditions and so on. Now you really have to evaluate and see, do I see a difference in passing and failing in these different stadia? So that is more up to you, and see what you can do depending on the device. And I think that's one case you had, Audrey, to discuss here to help them.
Yes, so we had a phthalate-free tubing set that we were evaluating. And so what we did is we tested multiple different, so all combined it was failing. So we tested the components again to find the cytotoxic component. And what we found out is there was one component that had a plasticizer in it. And so it may not have had the phthalate that they were saying that the whole system didn't have, but it had another phthalate, another plasticizer that would be captured in the cytotoxicity test. So again, this was information that they got from their supplier saying, oh, this is phthalate free. I think it just identified DEHP free, but there was an alternate phthalate used that we were able to pick up. So again, asking those questions, asking the suppliers so that we can get that detail listed. And then we could then understand what's the risk of the patient. Is the dose okay? Do we do specific testing? Do they find an alternate component? You know, you can ask more questions to get to a solution after that.
Yeah, I think that's what we have seen also within extractables and leachables testing. If you say this product doesn't, or in polymer manufacturing, if it doesn't contain a certain plasticizer, they're using something else. They add a plasticizer for the right reason. It has its reason why it's used. So not using it, it is replaced by something else, and that can indeed cause a failure. So it's something that happens a lot, not only with cytotox failures, it's something that we see often. So when we are here in this phase, we have looked at the lab, we have looked at the extraction condition, the materials, we have investigated now at our own site in our process. Now we are not randomly, but we're thinking, okay, what can we do? Some investigational testing. If we found a root cause and if we find a solution, then again, we're going to repeat ourselves. You have to go through the assessment. You're never getting out of this room without a proper risk assessment. So even the documentation of your investigational testing should be there with the information from your material investigation, and so on and so on. So it becomes a larger document. The longer you're in the room, I think, the larger the document gets.
Yes.
Yeah.
Yes.
The longer your story, the longer your team building. So you can see it in a positive or a negative way. I will see it in a positive way.
Yes.
So then you're out after step 5.
Yes.
We're one to go.
One to go. Let's see if we can find our last clue, which will prove to be our most difficult clue, but it's here on the computer screen. It will be data is what we're going to look at. So this is where you're most likely going to go straight to a risk assessment where all of these other situations just don't apply. You're at the end of your rope. Again, staying in that room a long time, but we still have an out. So this is where we want to be really thoughtful about the use of the device and what these results mean to a patient exposure. So it's important that we give cytotoxicity testing the weight of evidence that it deserves and can provide, but it's, again, not the end-all be-all. It's just the beginning point and tends to be the hardest gate to get through, to get through biocompatibility. So I want to give an example. So in this case, this needs to be a document written by experts who can analyze the device and understand regulatory expectations. So maybe that's a combination of a biocomp expert like myself, a toxicologist that we'll need, and a material expert. So just some ideas of combining all of that expertise together.
Teamwork.
So I want to give, I want to give an example of a case that we came up against where we had tested a full device and we got a failure. So we thought, okay, we're going to start our investigation. We're going to test each of the components separately. And in that case, we got a pass for each of the individual components, but combined, we couldn't find a cytotoxic component. There wasn't a lab error, nothing different in the testing, nothing in the processing. We went through all the steps and there wasn't anything that we could point to say, this is why we're seeing a cytotoxicity failure. But cumulatively, all together, we're seeing that. So the question really was, what's the patient exposure? And is there truly a risk here? Do we have other data that's more significant as far as if something toxic is coming off? Do we have other data to say, is there a biological response expected from whatever that is that's coming off? So we were able to pull all of that together to tell a story about patient safety. In this case, the device was incredibly low risk, had a low patient contact. Even at our lowest extraction parameters, we still weren't passing for a full combined device. So we were able to just tell the story one by one about patient exposure, possibility of risk, pull in the other testing data to support, in the end, patient safety altogether. So this is a more complex case, and I would say more rare. It is more rare that we come up against this. Usually we can identify a cause. Like Lisa said at the beginning, she was pointing out, you'll have latex, or you'll have a metal, or you have an antimicrobial agent in there. Those things we do expect to fail cytotoxicity, so we can walk through this a little bit easier. But even if you get to the point where you've checked everything and you're at the end, we still have an out with combined expertise to get this path forward approved at the very end. And then we have finally escaped. Lisa, we make a good team.
It's really, I think, the message is here, we can escape. We have escaped several times, and in the end, you can escape. Only, of course, the other data needs to support that, because if all other testing is failing, then yeah, you're not, of course, escaping. But to summarize, we closed our eyes today for a few seconds and we entered this beautiful room hidden or filled with clues that were hidden. We found them, all 6 of them. We solved the mystery of the medical device that has been extracted, the extract was exposed to cells, and we had a fail. That was our mystery, the cytotoxicity failure. So it used to be a burden. Now all of us can think, well, burden, it's something perhaps if we have the time. It's a great team building. It's a great way to investigate a lot of things. So that's, I think, our key message. And we gave you a 6-step approach where you start, of course, with the lab. You look at the testing conditions, then you look at the materials, then you look at your facility, at your processes. You may do some investigation, and in the end, for most of the time, you always need to write your story and what's the risk to the patient, because that's the most important thing. Look at your device. How is it in contact with the patient, and does it contain a potential problem? Is it the danger? Is it a risk? Yes or no? And with that approach, with step 1 to 6, if you follow that in the right order, I think it's very important, follow it in the right order, your story is shorter, you can really escape. So that's for us, for the talk today. If you want, and we have written this book, it's called it's a brochure or a white paper, stepwise approach. Actually, it should read How to Escape from a Cytotox Failure. So it's actually our escape room guidance. So if you're locked in this room, you can just download it from our website. I also want to say thanks to Kathleen Pammen, who actually co-created this document together with Audrey and myself. So it really helps you when you're in the room with your colleagues. We even have had some customers that say like, hey, we're at step 4, we're stuck, can you help us? So it's actually what we've been talking to. It actually guides you in these 6 steps, so you make sure that you follow them in the right order. And that's also how you play an escape room. Solve the clues, in the right order. That's how it's designed. So that helps. And it also helps us, I think, Audrey, to say, okay, we're at step 6, step 3. It helps us to talk with customers. So it also gives us some guidance in how to write our story and how to address it.
Yes.
So, and of course, if you're stuck in the room, you can read the document, but you can also like put like the emergency button, and you can or wave to the camera like, hey, I want to get out. You can contact me or Audrey. So here are our email addresses, so you can reach out to us, and we're more than happy to give you some tips once you're in the room and try to give you some clues so you can easily find your way out. And with that, we're going to end. I think it's very important to let you know that everything and this presentation can be found on our Zotero Academy. I think, Audrey, you have put already a lot of content with your team on this platform.
Yes, so I think it's great to highlight that, we want to support our customers as much as possible. So education is an important part of that. So Zotero Health has started an academy, and as Nelson Labs, we've posted a lot of information. And I've actually created a biocompatibility pathway, which is our first education pathway that we're launching. So if you want to learn more or find that you need to build your expertise at your company, hop onto this website, register. The content for right now is free. So you can get on there and see what we have available. And again, we will add this webinar at a later date to that academy.
Okay. Thank you very much, Audrey. It was very nice to play this game with you. And
thank you.
Let's create another room for the future and see if we can.
I agree. Good team building.
And thank you for listening.
Thank you.